Assalamu alaikum, this is SABAR Khan and the topic is two main types of electron microscope. The first one is the TEM, which stands for transmission electron microscope, and the second one is the SEM, which stands for scanning electron microscope. First we will come towards the transmission electron microscope. In transmission electron microscope there is a vacuum chamber. Vacuum is necessary because when any air is present, that air will interact with the electron beam, so the intensity of the electron beam become disturbed, and after that the clarity of the image is disturbed. So a vacuum chamber is necessary. This is a vacuum chamber. This is electron gun. These are the condenser lenses. This is the condenser aperture. This is the sample which is present on the sample stage. This is the objective lens. This is the objective lens aperture, selected area aperture. These two are the intermediate lenses. This is the projector lens, and this is the fluorescent screen. So electron gun emits the electrons. This is the electron beam emitted from the electron gun. The electron gun is mainly a tungsten element. So when the electron beam emits from the electron gun, this electron beam is condensed by the condenser lens 1, and when this electron beam passes from the condenser lens 2, this electron beam is condensed further. These condenser lenses are present to make the electron beam small, thin and coherent. So the small, thin and coherent beam then pass from the condenser aperture and come towards the sample or specimen. And in transmission electron microscope, the sample is very thin, so the electron beam passes from the sample and come towards the objective lens. But during passing this electron beam from the sample, the electron beam forms the image of the sample, and this image is magnified by the objective lens at some extent. This formed and magnified image passes from the objective lens aperture and selected area aperture and come towards the intermediate lens 1. This intermediate lens 1 magnifies the image at some extent, and when the image passes from intermediate lens 1 towards the intermediate lens 2, this intermediate lens 2 further magnifies the image, and after that it comes towards the projector lens, so the projector lens further magnifies the image, and after that the fluorescent screen is present, in which we can see the formed and magnified image of the sample. So this is the transmission electron microscope. The transmission electron microscope is called transmission electron microscope because in this type of microscope the electron beam transmit or pass from the sample. That is why it is called a transmission electron microscope. Now we come towards the scanning electron microscope. There is also a vacuum chamber, and this is electron gun, this is anode, this is the condenser lens, this is scanning coil, objective lens, sample, and this is secondary electron detector, and this is the screen. When the electron emit from the tungsten element or electron gun, this electron beam is accelerated by the anode, and this electron beam comes towards the condenser lens, which condenses the electron beam, and after that this electron beam passes from the scanning coil. Scanning coil is a type of coil which performs a raster scanning. Raster scanning is a type of scanning which scan the sample row wise. For example, this is the sample, and raster scans the sample like this. This is first row will be scanned, second, third, fourth, fifth, and so on. So the sample is scanned row wise. The scanning coil deflects the electron beam like this: first row, second row, third row, and so on. So this is the scanning coil. After that, the electron beam passes from the objective lens and comes towards the sample. The objective lens focuses the electron beam on the sample. This electrons which emits from the electron gun is known as primary electrons, and when these primary electrons come towards the sample that emits the electron from the sample, that are known as secondary electrons. And these secondary electrons are detected by a secondary electron detector, and an image is formed according to the secondary electron detector on the screen, and we can see the image on the screen. This is called the scanning electron microscope because the electron beam do not transmit or do not pass from the sample, but the electron beam scans the sample and emits the secondary electrons. That is why it is called scanning electron microscope. So this is all about the transmission electron microscope and scanning electron microscope. Now we come towards the difference between these two types of electron microscopes. Transmission electron microscopes provide two-dimensional image, while scanning electron microscope provides three-dimensional image. Here inside detail of the sample is provided, while here surface detail is provided by the scanning electron microscope. Here very thin sample is needed for the transmission of the electron beam, while here variable size of sample is required. Electrons pass through the sample. Here the primary electrons come towards the sample and emits the secondary electrons from the sample. This is more expensive. Transmission electron microscope is more expensive. Scanning electron microscope is less expensive. The speed is slower, its speed is faster. It has greater resolution while it has less resolution power. More time is required for sample preparation, while here less time is required for sample preparation. And its operation is more complicated, while its operation is simple and easy to use. So this is all about the transmission electron microscope and scanning